Let us see sexual reproduction in flowering plants. Flowering plants are also called as angiosperms. Where does actually the sexual reproduction occurs? It occurs in flowers. The two important structures we should know related to the sexual reproduction are male reproductive structure and female reproductive structure. Male reproductive structure is also called as androecium, and female reproductive structure is also called gynoecium. Have you heard of a stamen? Stamen is the structure which represents male reproductive organ, and pistil is the structure which represents female reproductive organ. These two organs play important role in sexual reproduction. At its core, what happens in reproduction? Stamen produces gametes, which are called male gametes, and pistil produces gametes, which are called female gametes. These two gametes should meet and fuse to form zygote. Zygote develops into seed. We know that seed develops into a plant. So, gametes from one plant or flower of one plant are fused to form a seed which results in the production of another plant called offspring of this parent plant. That is a simple saying of reproduction. Now let us see the story of this reproduction. First, we will study about the structure of the stamen, which is a male reproductive organ. Then we will study about the structure of a pistil, which is a female reproductive organ. Then we will see the interaction between them. Now, stamen. If you see a stamen, it has two parts, anther and filament. We are more interested in anther when we talk about reproduction. An angiosperm anther, that is, anther of a flowering plant, is bilobed. That means it consists of two lobes, and each lobe have a two theca. That's why they are named as dithecous. The anthers of angiosperms are dithecous. But one single anther will have four theca. But the name dithecous is given based on number of theca in single lobe. In summary, anther has two lobes and each lobe has two theca. Now we have to see the tissue makeup of this anther. To study the tissues of the anther, we have to cut anther in transverse section. When anther is cut in transverse section, its shape looks like tetragonal. That is, it has four sides. And the tetragonal shape also have four corners. At each corner, there is a specialized tissue called microsporangia. So, how many microsporangia are present in one anther? Four microsporangia are present, which are located at the corners. Each lobe will have two microsporangia. This specialized tissue, that is microsporangia, develops further to become pollen sacs. And inside these pollen sacs, we will see pollen grains. Now we will see the detailed structure of microsporangium and how it develops into pollen sac containing pollen grains. When you see a transverse section of a microsporangium, the shape is near circular. It is not complete circular. It is near circular. And this microsporangium is surrounded by four layers. We will see these layers from outside to inside. The first layer we encounter is epidermis. The second layer is endothecium. Third layer is middle layers. And the fourth is tapetum. What are the functions of these layers? The outer three layers, that is epidermis, endothecium, middle layer, perform a function of protection. Whom they are protecting? They are protecting developing pollen grains. And when time comes, these three layers dehisce, that is, they rupture, resulting in release of pollen grain outside. Now the fourth layer, tapetum, it functions as a nourishing layer. It nourishes the developing pollen grain. Nourishes means it provides food to the developing pollen grain. That's why if you see the cells of the tapetum, they possess dense cytoplasm because they have food material. And also they have two nucleus or more than one nucleus because they should be in continuous activity to provide nourishment to the pollen grains. That's why their cells are with dense cytoplasm and more than one nuclei. Now we will see anther at different stages of development. First, when the anther is young, at the center of the microsporangium, there is a tissue called sporogenous tissue. What is tissue? Tissue is a group of cells. So, this sporogenous tissue contains group of compactly arranged, That means there is no space between the cells. They are in touch with each other and they are homogeneous. Means they look similar. Now, I'll call these cells in this porogenous tissue as microspore mother cell or pollen mother cell or potential pollen. We will see why we are calling these cells with this name. Now, as this anther develops, the sporogenous tissue or cells present in the sporogenous tissue undergo meiotic division. Now, this is very important. Till now, they are diploid cells. With the meiotic division, we are getting haploid cells. So, pollen mother cell or micropore mother cell or a potential pollen is diploid in nature. They undergo meiotic division. When these undergo meiotic division, cluster of four daughter cells are formed. This cluster of four cells is called microspore. Why they are cluster? Because still after the meiotic division, they are together. The four daughter cells are together. The other name given for this is microspore tetrad. Now, as anther matures, when anther matures, dehydration occurs. That is, loss of water. And these microspores or the cells present in the microspore disassociate from each other. They get separated from each other and then they develop into pollen grains. So inside each microsporangium, thousands of microspores or pollen grains are formed and they are released outside. When they will be released outside? When the outer three layers or three walls of microsporangium undergo dehiscence. And this process of formation of pollen grains or formation of microspore tetrad from the sporogenous tissue by meiosis is called microsporogenesis. Now we understood how the pollen grains are formed in anther. Now we will see little detail about pollen grains. Pollen grains represent male gametes. Remember, now we got male gametes. If you take a shape of a pollen grain, they are generally spherical or round in shape. Generally, the other shaped pollen grains are also present, but most of the time they are spherical. And the diameter of this sphere is 20 to 50 micrometers. Now we will see what does the pollen grain contains from outside to inside. Pollen grain has a prominent two-layer wall. The outer wall is called exine and inner wall is called intine. The outer wall is hard, and this hard outer wall is made up of a chemical called sporopollenin. And the inner wall is not hard, it is thin and it is made up of cellulose and pectin. If you see, the outer wall is not continuous. At some places, there are holes, also called pores. These pores are called as germ pores. At the site of germ pores, sporopollenin is absent. But inner intine wall is continuous. Let us discuss little detail about sporopollenin. It is a organic material, and the important point of this organic material is it is one of the most resistant organic material known. Resistant means it can withstand high temperatures. That means high temperatures cannot destroy this sporopollenin. And even strong acids or strong alkalis cannot react with this sporopollenin. So, this property also makes pollen grains to be well preserved as fossils. Now, inside these two layers, there is a cytoplasm which is covered by plasma membrane. When pollen grain matures, it contains two cells at its center. There is one cell called vegetative cell and the other cell is generative cell. How these two cells are present is very interesting. Vegetative cell is a bigger, and this is vegetative because it has abundant food reserve, and the nucleus is also very large and irregularly shaped. Now come to the generative cell. It is small and the shape of the generative cell is spindle shape. This spindle shaped small cell has a dense cytoplasm and a nucleus. Where does this generative cell is present? This is interesting. Generative cell is present inside the cytoplasm of the vegetative cell. And what is it doing there? It is floating in the cytoplasm of the vegetative cell. See how fascinating it is. Now, we know that pollen grain is released from the anther. When it is released, if it contains one vegetative cell and one generative cell, then it is said that it is released at two cell stage, which is seen in 60% of the angiosperms. In rest of the angiosperms, what happens? The generative cell, which is floating, it undergo mitotic division. Remember which division? Mitotic division. So when it undergoes mitotic divisions, we get two generative cells. When pollen grain is released after the mitotic division, how many cells it has? Three cells. So it is said that the pollen grain is released at three cell stage. Now we will see. Now we got the male gametes or structures representing the male gametes, that is pollen grains. We will just deviate out of our story. How we human beings use these pollen grains or what they cause to us. For plants, they are gametes. They take part in the sexual reproduction. But for human beings, what is their role? Pollen grains causes disease in human beings. They cause severe allergies and lead to chronic respiratory disorders like asthma and bronchitis. And there is a vegetative cell which has abundant of food material. So pollen grains are rich in nutrients. So it has become a fashion in recent year that pollen grains are used as a tablets or food supplements. If you take in a Western countries, the products of pollen grains are sold as tablets and syrups. And other use is, it is found that when pollen grains are consumed, the performance increases. So they are consumed by athletes to increase their performance, or they are even given to race horses to increase their performance. Okay, now we will come back to our story. Pollen grain is released from the anther. Once it is released from the anther, it has to go and meet the female gamete. To meet the female gamete, it has to land on the female reproductive organ, which is pistil. Now, there is a time between the release of the pollen grain and its landing on the pistil. During that time, does this pollen grain is viable? If it is viable, for how much time it is viable? That is, how much time it has before dying to land on the female reproductive organ. It varies. In rice and wheat, it has to land on the female reproductive organ within 30 minutes. Otherwise, it will die. If you take members of Rosaceae, Leguminosae, and Solanaceae, they maintain viable outside for months. See the difference. For some plants, the pollen grains of some plants can live for only less time, but pollen grains of some plants can live for months before reaching their female reproductive organs. And pollen grains of large number of species can be stored in liquid nitrogen. What is the temperature of the liquid nitrogen? Minus 196 degrees Celsius. They can be stored at this temperature for years together. And such stored pollen grains are used as a pollen banks. Next, we will discuss about female reproductive organ called pistil. Pistil has three parts: the stigma, style, and ovary. From above down, they are arranged like a flask. At the top of the flask is the stigma. Bottom of the flask is the ovary, and the structure connecting both is style. The pollen grain, which is released from the anther, lands on stigma. It is passed through a long, slender structure called style. Then it enters a broad or bulged basal part called ovary. And if you see inside the ovary, there is a empty space. That's why it is called ovarian cavity. The other name for ovarian cavity is locule. Inside the ovarian cavity, there is a placenta. To this placenta attached is a megasporangia. Megasporangia attaches to the placenta. This megasporangia are commonly called as ovules. Now, does a flower contains single pistil or many pistils? Both are correct. A flower can have single pistil or it can have many pistils. When flower has a single pistil, it is called monocarpellary. And when it contains more than one pistil, it is called multicarpellary. When more than one pistil are present, that is multicarpellary. All pistils are fused together. In that case, it is called syncarpus. And when these pistils are separated from each other or free from each other, it is called apocarpus. Now we have discussed about the number of pistils. Come to the microsporangia. Sorry, come to the megasporangia. Megasporangia are also called ovules are attached to the placenta. A single pistil may have or ovary may have one ovule or it can have more than one ovule. Now we will see the detailed structure of megasporangium or ovule. Ovule is a small structure. We have seen that it is attached to the placenta. It is attached to the placenta by means of a stalk, a structure called stalk, which is called funicle. So, funicle is the structure that connects ovule to the placenta. And the part of the ovule which fuses with this funicle is called hilum. So, hilum represents junction between the ovule and the funicle. Now, if you see a ovule, it has a envelopes or protective walls or protective coverings. The protective envelopes of ovule are called integuments. There can be one integument or two integuments. Inside these integuments, there is a mass of cells. That structure is called nucellus. Now, integuments cover the nucellus all around, but at one part, the nucellus is not covered by the integuments. That small part is called micropyle, or we call it as a small opening. And opposite to this micropyle, it is also called as micropylar end. Opposite to this micropylar end, the region is called chalaza. So chalaza represents the basal part of the ovule. So micropylar end and the chalaza are opposite to each other. Now, I told nucellus as a mass of cells, and the cells of the nucellus have a abundant food materials. Why they should have food materials? Because they should be nourishing something which is coming soon, which is a embryo sac. The embryo sac is also called female gametophyte. Now we have reached to the female gametophyte, but we have not yet seen how this female gametophyte is developed. The development of female gametophyte is called megasporogenesis, and this occurs from megaspore mother cell. Oh, from where this megaspore mother cell has formed? Take, if you see a cells of the nucellus present in the micropylar region, one of the cell differentiate into megaspore mother cell. So one of the cell of nucellus differentiate into megaspore mother cell. So this cell is a large cell compared to other cells of the nucellus, and it contains dense cytoplasm and prominent nucleus. And this megaspore mother cell undergoes meiotic division, and this meiotic division results in the production of four megaspores. In anther, we got four microspores. Here we are getting four megaspores. If you see in majority of the plants, among these four daughter megaspores, only one is functional, while the other three degenerate. Compare it with the male counterpart. There, all four microspores develop into pollen grains, but here only one megaspore among four develops into embryo sac or female gametophyte. That's why this method of embryo sac formation from single megaspore is termed as monosporic development. Mono means single. So here the embryo sac is developed from single megaspore and other three megaspores degenerate. This is called monosporic development. Now we have one functional megaspore. This functional megaspore cell develops into embryo sac. Functional megaspore has two poles, one towards micropylar end of the ovule, other towards chalazal end. Let us call this as a central cell. Here, during the formation of embryo sac, changes related to two structures takes place, one with the nucleus and other with the cytoplasm. These changes does not occur simultaneously. First nuclear changes occur, then cytoplasmic changes occur. Total three mitotic divisions of nucleus takes place. Nucleus which is present at the center of the cell undergo first mitotic division, resulting in formation of two nucleus. They migrate or move to opposite poles, one towards micropylar end and other towards chalazal end. Now it is called two nucleate embryo sac. Further division of nucleus occurs at poles. Second mitotic division takes place, resulting in four nucleate stage. Third mitotic division results in 8 nucleate stage. Now, there are 4 nucleus at each pole. Now, cytoplasmic changes occur by formation of cell wall around nucleus. At micropylar end, cell wall gets deposited around 3 nuclei, resulting in the formation of 3 cells, 2 synergid cells and 1 egg cell. These 3 cells together called egg apparatus. Synergid cells also have end towards micropyle. Their wall at this end is thick and form filiform apparatus. Cell wall also deposits around 3 nuclei at chalazal end. Three cells at this end are called antipodal cells. Total, six nuclei are surrounded by cell wall. Now, two nuclei are still free at each ends. These two nuclei again move and situated below the egg apparatus. All other nucleus became separate cells after deposition of cell wall. These two nuclei are still in central cell. So, embryo sac has one large central cell with two nuclei, two synergid and one egg cell at micropylar end, and three antipodal cells at chalazal end. So, embryo sac has 7 cells and 8 nuclei. From previous discussion, we learned that pollen is a male gametophyte which has male gamete, and embryo sac is a female gametophyte which has female gamete. Now pollen and embryo sac has to meet for fertilization, but they are immotile. That is, they cannot move. Someone has to bring them together. The process of getting pollen, which is released from the anther to stigma of pistil, is called pollination. Just think you are in a garden of some flowering plant. There are many plants, and also there are many flowers in a single plant. Each flower has a stamen and pistil. Now see the possibilities. Pollen from one anther can land on pistil of same flower or pistil of other flower of the same plant or pistil of other flower of different plant. Based on this, we have autogamy, geitonogamy, and xenogamy. Autogamy. Here, pollination between stamen and pistil of same flower takes place. Here, we have to know about two types of flowers. One is chasmogamous flower. Here, both anthers and stigma are exposed to outside. For successful self-pollination, pollen is released when stigma is receptive, and both anthers and stigma lie close to each other. The other type of flower is clastogamous flowers. Here, flowers do not open at all. Here, anthers and stigma lie close, and pollen has to invariably lands on stigma. Plants like viola, oxalis, and commelina produce both types of flowers. Next is geitonogamy. It is pollination between anther of one flower and stigma of other flower of same plant. It is functionally cross pollination because it is occurring between different flowers. But as the plant is same, it is genetically similar as autogamy. Xenogamy. Here pollination occurs between flowers of different plants. It is only type of pollination that results in development of genetically different plant. It is an example of proper cross pollination. Let us talk about outbreeding devices. 1. Plants don't prefer autogamy or geitonogamy. The reason is inbreeding depression. Inbreeding depression is where chances of expression of recessive genes is more. Second, plants prefer xenogamy because xenogamy results in variation. But many plants produce hermaphrodite flowers. Hermaphrodite flowers are flowers where Both stamen, which is a male reproductive organ, and pistil, which is a female reproductive organ, are present in same flower. In this case, there is a high chance that pollen comes in contact with the stigma of same flower. This is called self-pollination. Self-pollination is not preferred by the plants. The reason we know, because it leads to the inbreeding depression. To prevent self-pollination, plants develop different devices. They are called outbreeding devices. The first device is the timing of pollen release from the anther and stigma receptivity. They are in non-synchrony. That is, pollen is released before the stigma becomes receptile, or stigma becomes receptile before the pollen get released. The second device is position of the anther and stigma. When they are at different position, it is difficult for the pollen to sit on the stigma of the same flower. Both these devices, that is non-synchrony and different positions, prevent autogamy. Third device is self-incompatibility. It is due to genetic mechanism. It prevents the self-pollen from fertilizing the ovules of the same flower. It occurs by two mechanisms. Suppose by any chance, if pollen comes and sit on the stigma of the same flower or of the same plant, the pollen germination is inhibited and also pollen tube growth is inhibited. Fourth device is production of unisexual flowers. There are two scenarios. If both male flower and female flower are present on same plant, then what happens? There is a geitonogamy possible but no autogamy. The examples of this kind of plants are castor and mace. In second scenario, the male flowers and female flowers are present on different plants. So in this case, autogamy is not possible, we know. And geitonogamy is also not possible because whole plant contain either a male flower or a female flower. Example is papaya. Let us know about agents of pollination. Agents of pollination help in transfer of pollen from anther to stigma. Wind, water, and animals are agents of pollination. Among them, wind and water are abiotic agents. Animals are biotic agents. Majority of the plants use animals as agents of pollination. If you take abiotic agents, that is wind and water, the pollination here is uncertain because there is a high chance of loss of pollens while transfer. To compensate it, the plants which depend on abiotic agents for pollination produce enormous pollen grains. Now we will see about abiotic agents. Among abiotic agents, wind is the most common agent. Here, you take a stamens. They are well exposed so that pollen grains are easily dispersed into the wind. And if you see the pollen grains, They are light, that is, they are weightless and non sticky, so that they can be easily carried by the wind currents. And now the pollen should land on stigma. Thus, plants which depend on wind have a large feathery stigma so that they can catch the incoming pollen grain. Usually, the flowers of these plants have single ovule and numerous flowers which are packed as inflorescence. Inflorescence help in trapping the pollen grains. One example is corn cob. Now, if you see water, water as a agent is used as a rare in flowering plants. Only around 30 genera, the two monocotyledons, use water as a agent. Examples include plants which present in fresh water are Vallisneria and Hydrilla. Plants which present in marine are sea grasses like Jostera. One important thing we have to remember here is all plants which are present in water doesn't use water as an pollinating agent. Some plants, even when they are present in water, they use wind and animals for pollination. Examples of such plants are hyacinth and water lily. Here, flowers emerge above the surface of the water so that wind and animals can act as an agent. If you see plants which use water as a pollinating agent, they produce pollen grains which have a extra protective layer that is made up of mucilaginous. This mucilaginous cover protect these pollen grains from wetting. Now, the female flowers has to receive the pollen grain. Two groups of plants are present based on female flower position. One group, like Vallisneria. Here, female flowers reach surface of the water. Male flowers or pollen grains are released in the water, and male flowers or pollen grains float passively by the water currents. Some of the pollen grains may reach the female flowers, resulting in fertilization. Other group takes sea grasses. Here, female flowers are submerged inside the water, and pollen grains are also released inside the water, and they are carried passively towards the female flowers. Here also, some of the pollen grains may reach the female flower. And one more thing, the pollen grains which are released inside the water are long and ribbon like so that they can float easily. Now let us see about animal pollinators. Pollination can be carried out by small animals or large animals. Small animals include bees, butterflies, flies, beetles, wasps, ants, moths, birds, and bats. Large animals include primates, rodents, and reptiles. Among small animals and large animals, pollination by small animals is common. Among small animals, the dominant pollinators are insects, particularly bees. Now there are two kinds of flowers. One kind of flower where pollination can be carried out by variety of species. That is, they allow variety of species to pollinate them. The other kind of flowers, they are specifically adapted for a particular species. That is, only one type of species can pollinate them. If you take a insect pollinated flower, these flowers are usually large, colorful, fragrant, and rich in nectar. They are large because the insects should see them. They are colorful and fragrant, have a good smell, so that insects are attracted. Suppose if the flowers are small, they cluster to form inflorescence so that insects can see them. Does every flower produce good smell? No. Some flowers produce foul smell or foul odors. They attract flies and beetles. And we have seen they are rich in nectar. Nectar is a reward. When insect is attracted to the flower provides reward because they help in pollination. What rewards a flower can give? The most important rewards are pollen grain and nectar. Pollen grains, which are pollinated by animals, are sticky in nature. Let us see why they should be sticky. Now take a one insect. That insect is attracted by colorful and fragrant flower. It goes to that flower, and in that flower there is a nectar. Insect consumes that nectar. During this process, pollen grains are dusted on the insect. So these pollen grains should be sticky to hold on with the insect. The same insect with pollen grain now visits flower in other plant. There also it feeds on nectar. Now this pollen grain lands on the stigma of that flower. So that's how animals carry out pollination. Let us see about other floral rewards. Flowers provide safe place to lay eggs. One of the example is the tallest plant, Amorphophallus. The similar kind of relationship exists between moth and yucca. Moth and yucca cannot complete their life cycles without each other. Moth deposits its eggs in ovarian cavity. In turn, the yucca flower gets pollinated. Here, the yucca flower gets only pollinated by moth. It cannot be pollinated by other species. It is like a one-to-one relationship. As larva comes out of the egg of the moth, the seeds of the yucca plant develops so that larva can feed on some of the seeds of yucca plant. So here yucca is providing place to lay eggs and also providing some seeds for larva to feed on. Now let us discuss about pollen pistil interaction. Imagine a pistil with its stigma. This stigma can receive different types of pollen. One type is compatible pollen, that is the right pollen, or the other type is non-compatible pollen or wrong type. When can be a pollen be wrong for this stigma? When this pollen is from other species or from the same plant. When right pollen sits on the stigma, then the post fertilization events are promoted. But when wrong pollen sits on the stigma, there will be a prevention of pollen germination, or it prevents the pollen tube growth in style. So here if you see, the pistil has a ability to identify which is a right pollen and which is a wrong pollen. This occurs due to continuous interaction between chemical components of the pollen and the pistil. So when there is a continuous interaction between the pollen and the pistil through their chemical components, the pistil can identify whether it is a right pollen for it or wrong pollen for it. Now let's see the events after the pollen lands on the stigma. So the pollen lands on the stigma. Once it lands on the stigma, this pollen grain germinates and it produces pollen tube through the germ pore. We have seen now the outer hard wall of the pollen grain as a pores called germ pores where sporopollenin is absent. Through this pore, pollen tube is produced. And the inside contents, what are the inside contents? Vegetative and germinative cells. They move into this tube. And this tube further grows through the style. Style is a slender stalk. Through that style, the pollen tube grows and it reaches the ovary. Remember, a pollen grain can be shed in two cell stage or three cell stage. If at all a pollen grain in two cell stage lands on the stigma, while passing through this pollen tube, it finishes its next stage, that is mitosis of the germinative cell, converting it into three cell stage. If already three cell stage pollen is landed on the stigma, then it passes through the pollen tube as it is. Now after the reaching the ovary, pollen tube enters the ovule. We know that ovule has a micropylar end and chalazal end. Pollen tube enters ovule through the micropylar end. Then it meets the synergid cells. We know that at the micropylar end of the embryo sac, there are two synergid cells and one egg cell. So this pollen tube enters the one of the synergid cell. And if you remember, we have seen there is a wall thickening of the synergid cell called filiform apparatus. So this pollen tube enters the synergid cell through the filiform apparatus at micropylar end. This events from the stigma till the synergid cells is called pollen pistil interaction. It is said to be a dynamic process because throughout its course there is a continuous interaction between the chemical components of the pollen and also the pistil. Now, if we understand this pollen pistil interaction in better way, like which chemical components are taking part and if we know exactly what are the chemical components, and if we could able to modify the chemical components, then we can control this pollen and pistil interaction. So, control of the pollen pistil interaction by human beings and producing the desired plants is called artificial hybridization. Here, artificial hybridization is used to get the desired characters from two plants. First, here we use desired pollen grains. Next, these pollen grains has to be landed on the female flowers, that is stigma, for further fertilization. The process of artificial hybridization is different depending on whether we are pollinating bisexual flowers or unisexual flowers. Let us see in bisexual flowers what happens. Now, one side we have desired pollen grains. These desired pollen grains should be used to pollinate the bisexual flower, stigma in the bisexual flower. Bisexual flower means in that flower we have a stigma and also stamen is also present. The stigma can receive pollen from its stamen only, that is self pollination, that has to be prevented. And this stigma can receive pollen from other plants also, that has also to be prevented, because we have a pollen grains, that pollen grains only should be used for this stigma. How can we prevent this self pollination? What we can do is we will just cut and remove the stamens. This process is called emasculation. So by emasculation, we are preventing self pollination. Done. Now, after that, we cover the flower with some bag. That is called, the technique is called bagging. So that when bag is put, the other pollens cannot come and land on the stigma. Then we wait. Once the stigma is receptive for the pollen, we open the bag and then dust the pollen grains we have. Then again close the bag to prevent the contamination. Let us see what happens in unisexual flowers. Unisexual flower means the female flower have only pistil. There is no stamen. So there is no risk of self-pollination. So emasculation is not required in unisexual flowers. But still, it can be pollinated by other pollen grains. To prevent it, bagging should be done. And same way, when it becomes receptive, the pollen grain of our choice can be used for pollination. So at the bottom line, bisexual flowers required emasculation and also bagging, and unisexual flowers require only bagging. Now let us talk about double fertilization. Why it is double fertilization? Because two fertilization events occur. You know, double fertilization is unique to flowering plants. Let us see about these double fertilizations. First, we have seen that the pollen tube has reached till the synergid cell. Inside the pollen tube, there are two generative cells. They are also called as male gametes. So, how many we have? Two male gametes. These two male gametes enter into the synergid cell. One male gamete move towards the egg cell, and the nucleus of the male gamete fuses with the nucleus of the egg cell. we know that nucleus of the male gamete is haploid and nucleus of the egg cell is also haploid. And this fusion is called syngamy. The fusion the fusion results in formation of a zygote. So zygote is a diploid and this further develops into embryo. So one fertilization is over. Now there is another male gamete. This male gamete move towards the central cell. Do you remember? Below the egg apparatus, we had two polar nuclei. So this male gamete move towards this polar nuclei. So male gamete has one nuclei and polar nuclei are two. Total there are three nuclei. All three nuclei are haploid in nature. When they fuse, all three nuclei fuse, it leads to the formation of primary endosperm nucleus. This primary endosperm nucleus is triploid. As there is a fusion of three nucleus, this is called triple fusion. And this central cell, which contains the primary endosperm nucleus, is called primary endosperm cell. It develops into endosperm. So this is the second fertilization. First fertilization is between one male gamete and egg cell, results in the formation of zygote, which later develops into embryo. The second fertilization is between male gamete and two polar nuclei, which forms a primary endosperm cell, which further develops into endosperm later. Now we will see post-fertilization events. Here we will study about endosperm development, embryo development, and how maturation of the ovule leads to the formation of a seed, and how maturation of the ovary leads to the formation of a fruit. Endosperm. If you see the endosperm development, its development precedes embryo development. That is, first endosperm gets developed, then the development of the embryo starts because endosperm provides nutrition to the developing embryo. The primary endosperm cell, which consists of triploid nucleus, this is a single cell. When this primary endosperm cell continuously divides, it is converted into the multiple cells. So the resultant structure is called triploid endosperm tissue. It is triploid because we know that it has a triploid chromosomes. And these cells are filled with food materials. Why? Because they have to provide nutrition to the embryo. Now, if you take pea, groundnuts, or beans, this endosperm is completely consumed by the embryo. So, if you see the mature seeds of pea, groundnut, and beans, endosperm is not seen because it is completely consumed. And if you see in a castor and coconut, this endosperm is not completely consumed by the embryo. So when you see the mature seed, still this endosperm is noticeable. Now we will see the common type of endosperm development. We have seen that the primary endosperm cell is converted into the endosperm tissue after division. How does this division took place? If you see many plants, there is a type of division. Now we will see that common type of endosperm division or development. Take a primary endosperm cell. Inside it, we have a primary endosperm nucleus. First, the nucleus undergoes successive division. After the successive division of nuclei, there are multiple nuclei in a single cytoplasm. They are called free nuclei. And at this stage, we call it as free nuclear endosperm. How many nuclei? It varies between the different species. Now, subsequently, that is after the nuclear division, the cell walls are laid. Then you see a tissue, like proper tissue. So this is the common type of endosperm development is seen. That is, first nuclear division occurs, then we see a laying of cell walls around it. Are you remembering something like this we have discussed before? Same in the development of embryo sac. First, the nuclear division occurs to form 8 nuclei, then the cell walls are laid. Same way, here endosperm also develops. If you take a coconut, the coconut water is nothing but a free nuclear endosperm. And the kernel, what we see, is a cellular endosperm. Now let us discuss about embryo. It develops at micropylar end of the ovule where zygote is formed. Early stages of development of embryo is same in monocotyledons and dicotyledons. Zygote develops into proembryo. Proembryo develops into globular, then heart shaped, then into mature embryo. Let us see the structure of dicotyledons embryo. Dicotyledons embryo has a embryonal axis and it has two cotyledons. Embryonal axis, which is present above the attachment of the cotyledons, is called epicotyl, and epicotyl ends at plumule, also called stem tip. Embryonal axis, which is present below the attachment of cotyledons, is called hypocotyl. Hypocotyl ends at radicle, also called root tip. This root tip is covered by root cap. Now, if you see and compare the monocotyledons embryo with the dicotyledon embryo, it also has embryonal axis, but here only single cotyledon is there. In grasses, this cotyledon is also called scutellum, which is present on one side and laterally. Embryonal axis, which is present above the attachment of this cotyledon, is called epicotyl, same as in dicotyledon's embryo, which ends at shoot apex. Here, this is covered by leaf-like primordium, which is called coleoptile. Embryonal axis below ends at radicle, which is covered by the root cap, which is in turn covered by the undifferentiated tissue called coleorhiza. So coleoptile and coleorhiza are seen in monocotyledon embryo. Now let us discuss about seed. Seed is said to be a final product of sexual reproduction. It is also described as fertilized ovule. It is present inside fruit. If you see the structure of the seed, it contains seed coat, cotyledons, and inside embryonal axis. Now, we know that what is endosperm. In mature speed, in mature seeds, if the endosperm is completely consumed, like in pea and groundnuts, they are called non-albuminous or ex-albuminous seeds. If residual endosperm is present, then they are called albuminous seeds. Examples are wheat, maize, barley, and castor. If you remember, the endosperm is surrounded by nucellus in ovule. Usually, nucellus doesn't present in the mature seed. If at all residual nucellus is present, it is called perisperm, which is seen in black pepper and beet. Remember the structure of the ovule. Ovule is covered by the integuments. These integuments form hard seed coat which covers the seed, and the micropylar end of the ovule present as a small opening in the seed coat. Through this opening, oxygen and water enters during germination of the seed. Mature seed loses some amount of water. There is 10 to 15% of water loss. And embryo is present as a dormant. That is, there is reduced metabolic activity. Under favorable conditions, the seed germinates in presence of oxygen, moisture, and suitable temperature. We know that ovules develop into seeds and ovary into fruits. These two events occur simultaneously. Wall of the ovary develops into wall of the fruit, which is called pericarp. There are fleshy fruits also and dry fruits also. Let us see what is true fruit and what is false fruit. When fruit is developed only from the ovary, it is called true fruit. Most of the fruits are true fruits. If other structures also contributes to the fruit formation, then they are called false fruit. Examples are apple, cashew, and strawberry. In these fruits, thalamus contributes to the formation of the fruit. We know that the fruit is developed after the fertilization. The same thing is true in most of the fruits. When fruits are developed without fertilization, then it is called parthenocarpic fruit. The example of parthenocarpic fruit is banana. We know that the production of this seed is a evolutionary advancement. There should be advantages of forming seed. So we will see pollination and fertilization in angiosperm plants doesn't depend on water. So even when there is no water, seed formation is expected. This is said as seed formation is more dependable and they are better adapted for dispersal and spread to the newer habitat. The seeds, the cotyledons of the seeds have enough food reserve. So it provides enough food to the early developing embryo until the embryo itself start producing food from photosynthesis. And the hard seed coat which is present, it also protects the early embryo. And the seed is produced by fertilization. So there is a genetic combinations. We know that when there is a genetic combination, variability is present. So which is necessary for development of new varieties. Seeds form a basis of agriculture. The important property for this purpose is dehydration and dormancy. Because of dehydration, they can be stored and used as a food. They are dormant, but when season comes, they can be used to raise the crops. Agriculture is all about food and raising the crops. So, seed is a basis of agriculture. Viability of seeds. How many days a seed can be viable? In some species, seeds are viable only for few months, and in many species, they live for several years. Even some seeds live for hundreds of years. Oldest seed known is from lupin, which is called Lupinus arcticus. This is excavated from Arctic tundra. This seed germinated and flowered after 10,000 years. Recently, another seed is excavated from King Herod's place. It is of a date palm, that is Phoenix dactycifera. It is 2000 years old seed. Now let us discuss about apomixis and polyembryony. We know that the seeds are produced by fertilization. They are said to be products of fertilization. But a few flowering plants like Asteraceae and grasses produce seeds without fertilization. It is called apomixis. When a fruit is produced without fertilization, it is called parthenocarpic fruit. Here in apomixis, seed is produced without fertilization. So basically, apomixis is a form of asexual reproduction, but it behaves like or mimics as a sexual reproduction. There are several ways of development of apomictic seeds. In one way, in some species, the diploid egg cell is formed. If you remember what we have studied, the egg cell formed was haploid. In some species, the diploid egg cell is formed. We have seen previously the egg cell formed in the embryo sac was haploid, but in some species, diploid egg cell is formed and without reduction division, it develops into embryo. So, there is no requirement of fertilization. The other method is, which is seen in many citrus and mango varieties. You remember nucellar cells, no? Nucellar cells surround the embryo sac. These nucellar cells start dividing and protrude into the embryo sac and they develop into the embryos. So the embryos developed by the nucellar cells are diploid and the embryo developed from the egg cell of the embryo sac is haploid. So such species contain both types of embryos. And occurrence of more than one embryo in seed is referred to as polyembryony. Nowadays we see many hybrid varieties of food and vegetable crops are being extensively cultivated because these hybrid varieties tremendously increased productivity of the food. But there is a problem with the hybrid. When hybrid seeds are produced and they are cultivated, the progeny will segregate and do not maintain hybrid characters. That is, genes in progeny are segregated and do not maintain the characters, hybrid characters. So, the hybrid seed should be produced every year. This increases the cost of the hybrid seeds. So, all farmers cannot offer these hybrid seeds. So, if at all hybrids are produced by apomictic seeds, just think apomixis means there is no fertilization. No fertilization means no gene segregation. So, characters are maintained at every generation. So, there is no requirement of purchasing seeds every year. So, it is cost effective. Now, research is going on to produce apomictic seeds.